Good morning, class. In today's lesson, we are going to develop our understanding of the concept of genetic drift. Genetic drift is one of the three mechanisms of evolution, with natural selection and mutation having already been covered in class. So who can remember what mutation is? Alison?
A mutation occurs when DNA in a gene is changed in such a way as to alter the genetic message carried by that gene. This can either be a positive change for the organism, neutral, or it can be detrimental.
Yes, that's right. Now, could someone please give me a definition of natural selection? Yes, Irene?
Natural selection, that's the process whereby organisms that are better adapted to their environment tend to survive and they produce more adapted offspring.
Yes, that's correct. So now on to our new topic. Genetic drift is the change in the frequency of an allele within a population due to random selection from one generation to the next. Random selection means that this occurs purely by chance or accident. It is not due to one allele being more advantageous to the organism, because as we know, this would be natural selection. And for those who have forgotten what an allele is, it is an alternative form of a gene which accounts for a specific trait, such as eye colour, which is the trait or allele type we are using today in our experiment. A population's allele frequency is the fraction of the copies of one gene that share a particular form. The effect of genetic drift is larger in smaller populations and smaller in large populations. Vigorous debates wage among scientists over the relative importance of genetic drift compared with natural selection. Ronald Fisher, the famous evolutionary biologist and founder of population genetics, held the view that genetic drift plays, at most, a minor role in evolution, and this remained the dominant view for several decades. In 1968, Moto Kimura rekindled the debate with his neutral theory of molecular evolution, which claims that most changes in the genetic makeup of organisms are in fact caused by genetic drift. The process of genetic drift can be demonstrated by using 8 M&Ms in a jar to represent 8 organisms in a population. Consider this jar of M&Ms as the starting population. Half of the M&Ms in the jar are red and half are blue, representing 2 alleles for eye colour in the population. In each new generation, the organisms reproduce with random success. To represent this reproduction, we randomly select an M&M from the parent population jar and deposit that colour M&M into the next generation jar. The selected M&M is then returned to the original jar. We'll repeat this process until there's 8 new M&Ms in the second jar. So first off, we've picked a red M&M representing a red allele. Red one again. A blue representing blue eye colour. The third red. 2 blue. Another red. Another red. And the last red. So after 8 random selections, we've ended up with 6 red alleles and 2 blue. This new jar now contains a second generation of offspring, consisting of 8 M&Ms of various colours. Unless the second jar contains exactly 4 red M&Ms and 4 blue M&Ms, a random shift occurred in the allele frequencies. So, in our second generation, we have 6 red and 2 blue. So we'll repeat this process again, but now selecting from our new second generation with 6 and 2. First one's red, representing your red allele. Another red. A blue. red, red, 4 red and 1 blue, red, red, and for our last one, another red. So, after those 8 random selections, we've got 7 red alleles and 1 blue allele. This is continuing with our previous pattern of genetic drift. Now for our 4th generation. We'll choose randomly 8 new M&Ms from our 3rd generation. First is red. Red again. It's 3 red. So far we've all got red. Another red. Seven red alleles. And an 8th red allele. So, by our 4th generation, the red allele is fixed in the population and the blue allele has become extinct. This is all because of genetic drift. Just because from our first, in our first generation, we had a 50-50 population, and slowly after 4 generations, the red allele has become purely dominant over the blue allele. Through this experiment, we've seen a population go from 50% of red alleles and 50% of blue alleles to 100% red allele, where it's fixed in the population, and the blue allele does not exist. This has happened all through chance and luck. It's not to do with whether one allele is stronger than the other, it's just because a 50% chance of me drawing a red or a blue allele out of a container to begin with, and then the next generation is more likely to be a red since I had 6 to 2 ratio. So this concludes our lesson on genetic drift. Hopefully you've learnt and understood how simple it really is and how much of an impact it does make on evolution.